We are first going to start, as we typically do, with a little update on our future talks that we'll have in the seminar series. Today it's Dr. Aiko Iwasaki. And if we could have the next slide. We'd like to inform people about the Long COVID Symposium that's happening in a couple weeks on Tuesday, February 1st at 4 p.m. And you can see there the RSVP announcement. We are getting I'm getting announcements from everyone. I hope that many people can join us. And then next slide, please. Here is the agenda for this Long COVID Symposium featuring the following people. A lot of experts from around Seattle and Washington State that will talk to us about long COVID. Sounds like an interesting thing. And our next viruses and vaccines series will be on Tuesday, February 20th, with details coming soon. With that, I'd like to introduce Pavitra Roshaduri, who is a research assistant professor in lab medicine and pathology and our genomic expert and explainer. And if you could take over, that would be great, and talk to us, give us a little update, please.
All right, thank you, Jen. Is everybody seeing my screen okay?
Yes, looks good.
Great. All right, so I'm here to provide the quick, like 5 minutes genomics update. I'll keep it really brief because there isn't a whole lot to update you on since the last time, because we are still in the middle of our winter wave as it is. So when we look at wastewater data, this is from BioBot Analytics, what we're seeing is that wastewater levels are at their highest since the BA1 wave back in the winter of 2021-22. There are a couple of caveats to note that wastewater concentration does not directly correlate with cases, translate to case numbers, but it does often help in predicting a wave to come or case numbers to come. Also, something to note is that participation of wastewater sites and methodologies have changed over time. So there's a little bit of a grain of salt to take with this, but it definitely indicates that our wastewater concentrations are higher than this time last year from the XBB 1.5 wave, and definitely higher than our summer EG5 wave as well. A better metric sometimes is the hospitalization numbers, as well as the number of ED visits where there's a diagnosis of COVID-19. So these numbers are also up, but they are at similar, actually slightly lower than levels from this time last year. This is data from the CDC. Closer to home, so our lab, UW Virology, continues to do PCR testing. We receive about 500 samples per day, primarily from our hospital system. And this is both inpatient and ED visits. And what we are seeing often reflects what's happening in our area. And what we are seeing a slight increase in positivity rates, but it's small, 4 to 7%, as opposed to 1 to 3% earlier this year. And also all of our positives that have a CT below 31 are being pulled aside for sequencing so that we have a good understanding of what variants are also circulating in our region. Now, at the last genomics update, or actually 2 genomic updates ago, Trevor had this figure where he showed relative growth rates, so growth advantages of the different lineages that are circulating or have circulated prior, and how they compare in their growth rates compared to XBB 1.5. And he highlighted that the JN1 variant, which was, or rather, initially the BA 2.86 variant was one that was being monitored because it had this large number of mutations in spike. And that one, simmered for a long time, but didn't really take off. However, that lineage acquired another mutation in spike, this L455S, and that's been labeled as JN1. And Trevor showed in November that this had a growth advantage that was, among the fastest compared to any of the other lineages that were being tracked at the time. And sure enough, that lineage has taken off and is now thought to represent the majority of cases currently across the US. So this is data from the CDC's variant tracker, and you can see that it's estimated to represent over 60% of cases around the US. However, as we saw in the prior slides, this hasn't really driven a surge in hospitalizations so far. So really, we're in a kind of wait and watch situation with this lineage, in that we're going to probably now see that lineage diversify. We're already at over 15 named sublineages of JN1 that are currently being tracked. So it's the first lineage in a long time where it's actually taken off. For a while, we had a whole soup of different variants. No specific variant had really this kind of growth advantage. So it's definitely one that caught everybody's attention. So now we have to wait and see how it further diversifies, or if there's going to be another lineage that has a different combination of mutations that will replace this one. And final slide is really that besides COVID-19, the other respiratory viruses are also leading to people ending up in the ER and in hospitals. So we're also looking at flu and RSV numbers. This is pulled from ResVNet, the dashboard from the CDC. And although the COVID numbers for 2023-24 for hospitalizations are lower than this time last year, influenza and RSV-related hospitalizations are actually at or higher numbers compared to this time last year. And the wastewater data also shows that we're not quite at that peak yet. And that is all I have. So I will stop sharing and hand it over to Dr. Iwasaki.
Actually, I will introduce her first, and then
right. Sorry,
but thank you, Pavitra. That was really nice, a nice update. And I just want to say, as a pediatrician, our hospital is still so full of RSV that we don't have time for much else. So with that, I'd like to introduce our featured speaker today, Dr. Aiko Iwasaki, who is the Sterling Professor of Immunobiology and the Director of the Center for Infection Immunity at Yale University School of Medicine. She's also a Howard Hughes Medical Institute investigator and really is at the forefront of several long COVID investigations. Her research focuses on the mechanisms of immune defense against viruses at mucosal surfaces, which are a major site for entry for infectious agents. Her team currently is focusing on long COVID through involvement in multiple collaborative studies, including cross-sectional cohorts with Mount Sinai, a longitudinal cohort of Yale, and the Yale Recovery Study of the impact of vaccination on PASC with Dr. Krumholz. And with that, I'd like to introduce her. And Akiko, you can tell people how you'd like to handle your questions. And we really have until the top of the hour to handle all your talk and all the questions. So thank you.
Thank you so much, Jen. It's a real pleasure for me to be here. And I see some familiar names, and I wish I could see you in person. Anyway, I'm excited to present our work on long COVID. Let me share my screen. Oops, okay. Okay, so I hope you can see this. I'm gonna present, yes?
It looks good, thank you.
Okay, thank you. So today I'd like to present our ongoing work on trying to investigate the immunological mechanisms that underlie long COVID. And if you have urgent questions or any questions that needs clarification during my talk, please raise your hand or ask questions. All right, so before I go into long COVID, I'd like to emphasize that there are many other infections in the past that have led to similar types of chronic outcomes. And these are, many are these medically unexplained post-acute phase of symptoms that occur after infection with a variety of viruses. SARS-CoV-2 is the most recent one to be added to this list, but there are other ones: Ebola, dengue, polio, SARS, chikungunya, ABV, and many others, as well as non-viral pathogens, bacterial and parasitic pathogens that have been found to lead to similar outcomes. So I think this is an important thing to keep in mind when we think about the pathogenesis of long COVID, as it may not be specific to SARS-CoV-2. And what is the sort of underlying and overlapping mechanisms that results in these very similar outcomes of post-acute infection syndrome that we call PAIS. That's something to keep in mind when we are thinking about immunophenotyping. The other thing to mention is that this review, I had the pleasure of writing with Jan Chotka, who himself is a patient with an ME/CFS. And throughout the pandemic, I've learned to really, engage with patients as they are really the expert of their own disease. And so this is one of many studies that I have been able to co-author with patients and learn so much from their experiences. And throughout the talk, I'll try to highlight patient involvement in all of this. So based on the sort of underlying immunological underpinning of these PAIS, some of the hypotheses that we are pursuing, as well as many others, is just sort of root cause hypothesis listed here. One is that the viruses, even though SARS-CoV-2 is thought to be acute respiratory virus, it is now well understood that there are viruses, antigen and RNA or a combination thereof, found throughout the body months or years after the infection, even in seemingly asymptomatic individuals. So it's possible that there is viral reservoir established somewhere in the body that's continuing to stimulate innate immune responses and or adaptive immune responses against the antigen, and that could be triggering these types of syndrome. Second hypothesis is autoimmunity. It's possible that during the acute phase of infection, lymphocytes that are either bystander or overlapping with molecular mimicry between the virus and the self-antigens may be triggered, and this autoimmunity could be underlying some of the subsets of long COVID. Dysbiosis of microbiome, as well as reactivation of latent viruses like the herpes virus family members, may be another one. And tissue damage that's seen throughout the body, even after a mild COVID case, that may also be underlying symptoms that are specific to the organs that are being affected. And so long COVID, even though it's considered a disease, it's really a collection of multiple endotypes of disease. Could be 3 to 4 different endotypes that are underlying the disease. And in order for us to develop therapeutics that are successful, we need to understand the biomarkers as well as underlying causes of these each of these endotypes. And I think that will be the only way to really significantly help the patients. So we've also been able to collaborate with Professor Michelle Monge at Stanford to look at the impact of mild COVID in the CNS, chronic changes that occur in the CNS. And there we've noted many different changes that occur 7 weeks after a mild acute COVID infection in the mouse model, where we've seen reactive microglia elevated, as well as reduction in oligodendrocytes and myelination of axons in the brain. And this is happening without any detectable viruses. So I think now we are really trying to model each of these endotypes using animal models, drawing some causality between these types of measurements that we're making in humans. And of course, this is one of the most influential paper that was published earlier by Dr. James Seeth's group, where they looked at longitudinal investigation of COVID patients over time within the first 2 to 3 months of disease and identified 4 past anticipating risk factor, type 2 diabetes, SARS-CoV-2 RNA-emia, Epstein-Barr virus viremia, and autoantibodies. And this sort of really fits nicely with the 4 different hypotheses of root causes that I just described. But we're seeing evidence for a lot of these things emerging as we go further into the research in the pandemic. So as already introduced by Jen, we are investigating several different lines of studies in human clinical cohorts. The one that I'd like to describe today is the Mount Sinai Yale cross-sectional long COVID cohort study. Now we are adding ME/CFS participants to this. But so I will talk about this in detail today. But the other things that I want to highlight, one is the Yale Recovery COVID study, which we just uploaded onto medRxiv. If you're interested, follow me on Twitter at Viruses Immunity. I just did a Twitter thread on that, where we are finding features of immunological features that are associated with improvement after vaccination versus those who don't improve. The third cohort is the Yale Lesson Study. This is getting larger and larger. It's over 2,000 participants now. We are recruiting patients online to share their history, medical data, history, their prescriptions, their experiences throughout the long COVID journey, to learn from the patients what is helping them, what is harming them, and how do we learn about these endotypes through surveys, as well as biospecimen collection. And we're also looking at the same issues in post-vaccine syndrome. We have also a medRxiv preprint on this as well, if you're interested. And then we have the long impact cohort. So impact is a cohort that we established during the 2020, where we recruited the first patients that were coming into the Yale New Haven Hospital. We've done a lot of immunophenotyping to try to understand the acute severe cases of COVID, immunology behind that. But we're now following these patients over time and trying to understand what is predictive of their development of long COVID. So today, I'd like to focus on the first cohort study, which is the Mount Sinai Yale long COVID study. And this is in close collaboration with Dr. David Petrino at Mount Sinai and his team, Jamie, Laura, and Dana listed here. We've been working for since the early days of the pandemic together to try to understand the long COVID and its immune phenotype. And the Yale team listed here also contributed greatly to this study. Joan Klein is the first of the multiple co-first authors who are listed here, Rahul, Pei Wen, Sasha, Jeff, and Jillian, who contributed their expertise to this study. And Aaron Ring and David Van Dyke are senior co-authors of this paper, who contributed the rapid extracellular antigen profiling, as well as computational methods to analyze the data. So this study recruited participants who had either long COVID or who had recovered, the convalescent control, or who had never had COVID, the healthy control, as well as healthcare workers at Yale who donated blood and a survey for vaccine studies. And we also had an external long COVID cohort for validation of certain parameters. From these participants, we collected electronic medical records, symptom surveys, flow cytometry to analyze in real time their cellular composition, activation phenotype. We did the rapid extracellular antigen profiling, which is Aaron Ring's invention, SARS-CoV-2 antibody profiling, peptide display library. This is a Serimmune technology that we applied, as well as plasma proteomics. And we did this in a cross-sectional cohort. The age and sex were as much matched as possible. The long COVID participants are listed in purple here and convalescent control in orange. Acute severity of disease was mostly on non-hospitalized mild cases. We did this because we think that the severe COVID patients that received ICU treatments may have a different sort of underlying etiology of disease compared to people who develop it from mild COVID cases. Days from acute COVID-19 hovered around 400 days plus or minus. So this really is one of the sort of longest durations for long COVID studies that we've seen so far. Jumping into some of the features that were distinct in people with long COVID, we found that, for example, flow cytometry analysis indicated increases in non-conventional monocytes, as well as reduction in the circulating levels of CDC1. These are the dendritic cell, conventional dendritic cell type 1 that's critical for inducing cellular immunity. We also see elevated activated B cells and double negative B cells. In the T cell side, we see that circulating level of central memory T cells, the CD4 T cells, were slightly reduced, and then the exhausted T cells in both subsets, CD4s and CD8s, were elevated. And long COVID patients also had a strange set of cytokines that the T cells were secreting. For instance, looking at the CD4 T cells, we see elevation of interleukin 2, 4, and 6 that's increased from people with long COVID. And we also see that there are some T cells that are double positive for IL-4 and IL-6, which are pretty much only found in patients with long COVID and not other groups. We also see elevated levels of anti-spike IgG that's found in people with long COVID compared to convalescent control or healthy control. Now, this particular analysis, we only included participants who had 2 doses of vaccination, just to make sure that we're comparing apples to apples. So this elevated levels of anti-spike protein is seen in other groups as well, and this may indicate persistent antigens such as the spike protein in people with long COVID. We don't know exactly what this is representing. Next, we looked at plasma levels of various factors that we identified, and indicated in red circles are the factors that were distinctly elevated or reduced in long COVID. So the ones that are elevated are complement C4b, several different chemokines and cytokines, whereas there's one factor that was very uniformly reduced in long COVID patients, which was the circulating levels of cortisol. And when we looked at the Z-score for the cortisol, we see that the long COVID participants from Mount Sinai really had about half of the level of the cortisol that is found in healthy control or the convalescent control. Because of this striking phenotype, we also recruited an external cohort of long COVID patients. This is a Yale Pulmonary Clinic where they are seeing people with pulmonary issues with long COVID. And from that participant, also, we see a significant reduction in the morning cortisol level. And the time of collection of the blood was similar between these cohorts. So when we looked at this data, we thought something about the control of the cortisol secretion from the adrenal cortex may be impacted in people with long COVID. And what we wanted to understand is whether there is any central regulation of cortisol that's being impacted in people with long COVID. So looking at the hypothalamus, pituitary, and adrenal axis, we know that centrally the hypothalamus controls the secretion of adrenocorticotropic hormone, ACTH, secretion from the pituitary, which then act on the adrenal glands to secrete the cortisol. And so the cortisol itself would have a negative regulatory effect on the hypothalamic and pituitary secretion of these hormones. So we wanted to see upstream of the cortisol at the ACTH levels, and the ACTH levels were not significantly different between the 3 groups, indicating that perhaps there is a deficiency in the central control of cortisol, either the hypothalamic secretion of CRH or the pituitary secretion of ACTH, that is resulting in this reduced levels of cortisol. So now, since this is a single time point measurement, we're now extending this analysis to look at throughout the day, 48-hour measurement of cortisol from participants with long COVID. to see what the diurnal levels of cortisol looks like in these participants. Next, we looked at reactivation of latent viruses. We haven't done any microbiome analysis, but we started looking at evidence for whether there is any reactivation of latent viruses. And this is in collaboration with Aaron Ring on the REAP. What we discovered was that there was an elevated level of IgG against some of the lytic proteins from Epstein-Barr virus and VZV-GE. You can see that long COVID participants had elevated levels of these antibodies, whereas many other viruses that we've tested, there was no differences in the antibody levels. And this elevation is despite the fact that a similar percentage of the people were latently infected. So late antigen reactive antibodies were not different between controls and long COVID. So this is sort of illustrating that there may have been a recent reactivation of EBV and VZV in the participants with long COVID. Looking at the REAP score, as well as another measurement that we did with a peptide-specific reactivity to EBV GP42 with Cerimune, what we noticed that the participants with long COVID, again in purple, had significantly increased levels of IgG against P23, GP42 peptide, as well as VZV-GE. And curiously, the level of these antibody against EBV corresponded with the amount of these CD4 T cells secreting the double cytokine IL-4, IL-6. Whether there's any causal relationship between EBV reactivation and the features that are seen in the CD4 T cells, we don't know yet, but it's something that we're investigating. So what about autoimmunity? We also looked at using REAP, autoantibodies against 6,000 plus autoantigens, human exoproteome, and found that while many people did have autoreactive antibodies, the number of autoreactive antibody per participant was not different among the healthy control, convalescent control, and long COVID. And it did not correspond to the long COVID severity score. And so overall, using machine learning, we've identified key immunological features that can distinguish people with or without long COVID to 94% accuracy by using a handful of features, cortisol being one of them. So this is lower in long COVID participants. The other features that were identified by the lasso approach was the CD4 TCM and CDC1 levels that are being reduced in people with long COVID, as well as galectin and EBV GP42 reactive antibody being elevated in people with long COVID. So this really illustrated that there is something biological that underpins the long COVID pathogenesis, and that the psychosomatic explanation of this disease does not need to be invoked. Now, taking the same set of data, we further identify sex differences in long COVID. This is not even on medRxiv yet, but I asked permission from the first authors to share this data. Julio Silva and Takahiro Takahashi have been working very hard on trying to understand sex differences. So long COVID is more predominantly seen in women of ages between 30 to 50. That's sort of the highest risk demographic group for this disease. Of course, anyone of any age can get long COVID, but that seems to be impacted the most as the women of that age group. And we know that many autoimmune diseases are predominantly found in women. So based on this, we wanted to analyze the same data set by now dividing them into male versus female and control versus long COVID, and applying the same set of analytical tools that we've employed in our first study. So overall, what we found is that female with long COVID, so all the female data are in blue and male data are in red. So please forget any stereotyping about these colors. The females with long COVID had more symptom burden and more organ system involvement than the male counterpart. Looking at the distinct symptoms that are reported by these participants, we also find that while many symptoms are equally reported between male and female patients, such as fatigue, nausea, tinnitus, and so on, there are some female dominant symptoms that are in the bottom here. That includes dizziness, body temperature issues, cough, throat, chest pain. Those issues are more dominantly reported by female, whereas the most male dominant reporting was found for sexual dysfunction, and hair loss was the most female dominant symptom that was reported. So just by looking at this graph, one can sort of imagine that the disease pathogenesis and the organ types that are being affected might be quite different in male and female patients. So looking further into the immunological features, we see that certain features that were reported in the first study, such as the CD4 T cell exhaustions or the alpha IL-6 double positive cells, these were mainly found in female patients with long COVID, as well as this EBV reactive REAP score that we reported in the first paper. So EBV reactivation, as well as this strange IL-4, IL-6 double positive cells, which I told you are correlating with each other, are mainly the feature of female patients. We also saw with REAP some autoantibodies that were elevated against different regions of the brain and gonad tissues. Long COVID males, on the other hand, have elevated NK cells, TGF-beta2 and APRIL. And when we examined the hormones that were measured in a study, and asked which of these hormones are most predictive of long COVID, what we found is that the testosterone level, which was lower in patients with long COVID compared to their counterpart, the control group, female, testosterone turns out to be the most significant factor, hormonal factor that can predict long COVID in female patients. Cortisol is, of course, another factor that's significant here, but the per unit odds ratio is really much more strongly associated with the testosterone level for long COVID female prediction. And male patients with long COVID, they had similar levels of testosterone compared to their counterpart. Conversely, when we looked at the estradiol level, that was significantly lower in patients, male patients with long COVID. And estradiol level per unit odds ratio prediction was the highest compared to other factors, including cortisol. So this is quite interesting because we're now seeing evidence of not only cortisol reduction, but sex hormone changes that are uniquely found in female and males. So this part of the new part of the study basically is showing us that female long COVID patients have more symptom burden and more organ system involvement and unique symptoms that are reported only or more frequently in female than males. There are distinct symptoms. Female long COVID patients have elevated exhausted CD4 T cells, cytokine secreting T cells, and EBV reactivation, and some autoantibodies, whereas male patients with long COVID have elevated NK cells, TGF-beta, and APRIL. And lower levels of testosterone were seen in females with long COVID and was the top hormone predictor of the long COVID status. Whereas conversely, lower estradiol levels was the top hormone predictor of long COVID in males. So taking all of these things together, one can hypothesize that, and I already told you about the hypothalamus pituitary axis being potentially disrupted for cortisol secretion in the adrenal glands. But now we're potentially seeing the same kind of dysfunctional impact of HP organ axis with gonadal hormones being dysregulated. And if you extend this to other organs and tissues, one can hypothesize that there may be other organ that's controlled by the HP organ axis that may also be dysregulated in people with long COVID. So this is something that we are looking into. We already know that people with long COVID have a significant changes in their muscle, the type of different fibers that are found, as well as their mitochondrial count and so on. So it may be that this is a central kind of dysregulation that may be affecting multiple organs, and that can potentially explain why there are over 200 symptoms that are being reported in people with long COVID. So coming back to this 4 root causes of a hypothesis, we are now seeing significant evidence from many different laboratories of viral reservoir and viral sort of persistence of antigens and RNA. We are also seeing several papers reporting autoimmunity, autoantibodies in people with long COVID. And we're seeing this elevated levels, particularly in female patients, as well as reactivation of latent viruses and tissue damage that's being found by multiple different groups using in vivo imaging techniques. So to tackle these types of hypotheses, we really need to perturb the system. And one way in which we are doing that is Paxlovid, a trial that we are conducting with Yale Listen Study with Harlan Krumholtz group. We are recruiting 100 participants who are randomized into placebo arm or 15-day course of Paxlovid. And we're measuring symptoms before, during, and after, as well as immunological parameters that I described to you today are being measured from these participants. And the idea is to be able to identify biomarkers that are associated with improvement from taking Paxlovid. And if that is possible, then we're really interrogating the first type of hypothesis, the viral reservoir. Replicating viruses, if they exist, can that be dealt with by Paxlovid? And if so, can we get biomarkers? Because the biomarkers for diagnosis is critical for sort of identifying patients that might benefit from a particular intervention. And for this one, we are hopeful to get insights into that. Even if 10% of the patients benefit, if we can identify what kind of biomarker they had prior to the Paxlovid treatment, I think that will go a long way. And we're obviously also interested in autoimmunity and dysbiosis, reactivation, and so on. So we're developing animal models to reflect each of these root causes and interrogating sort of cause and effect for these hypotheses. So I want to end here by thanking a huge number of people who contributed to the work that I was able to explain today and beyond, and many funders that generously support our work. So I'm going to stop sharing here and take any questions.
Thank you so much. And thank you for giving us a very interesting and thorough background. And we already do have a few questions on the chat. I'm wondering, Michael Davis, perhaps would you like to start out? Because you had a few questions. I'd rather have you give it than you ask them than me.
Right, everyone can ask them. There were 2 questions. As far as the cortisol levels, do you think that the lower cortisol were a cause or a consequence of long COVID? And is there any way to go back and query any of the patients to see whether or not they had lower cortisol levels prior to study start? And then the second question was, given the recent tie between EBV and multiple sclerosis, do you think we'll see a correlation between those with long COVID and MS moving forward?
Thank you, Michael. 2 very excellent questions. So the cortisol changes, yeah, we're very intrigued about that, whether it's a cause or effect of the long COVID. We do want to say that we are noting that many people with long COVID have sleep problems, and that may be impacted by cortisol, or that may also impact the cortisol level as their circadian rhythm is significantly disrupted. So, and the cortisol reduction that we're seeing, it's not really a level that they would have been detected clinically before the COVID pandemic. So there may have been underlying issues there, but it's hard to know what their levels were before the COVID. And the other is, so there is an interesting sort of papers. There are interesting papers from the ME/CFS world where they did a low dose corticosteroid treatment, randomized clinical trials for those. And while people benefited from a cortisone treatment, it wasn't sustainable because it really impacted the endogenous levels of cortisol. Long-term use is very difficult. So I think there may be path forward in interrogating these questions with trials, but it has to be done in such a way that it's low dose enough just to restore the physiological levels of cortisol. I think it's possible to do. The second question about the MS, yeah, that's something that's sort of why we're really kind of looking into this autoimmune phenotype. Many autoimmune diseases or incidence increase after COVID infection, SARS-CoV-2 infection. So we wouldn't be surprised to see elevation in autoimmune diseases after the acute phase of COVID. For the MS, it may take several years to develop, but that's something we're really monitoring in our cohort. We're also, as I mentioned, making animal models to be able to see if, say, autoantibodies or these autoantigens that are found in long COVID patients, whether that's sufficient to trigger autoimmunity. So lots to come.
Thanks. Perhaps Dr. Van Voorhees, Wes, can you ask your question?
Sure, thanks. That was a terrific seminar, Dr. Iwasaki. A couple of quick questions. Did you say that the Yale recovery study suggested improvement in long COVID neuro outcomes and COVID vaccination or revaccination? And then the second question is, how long is Paxlovid going to be given in the Yale LISTEN trial?
Yes. Okay. So Paxlovid will be a 15-day oral treatment. And we wanted to do it longer than 5 days because if there is persistent virus replicating, it's likely going to be replicating at a very slow pace. And we wanted to capture every cycle with Paxlovid, and that's why we're doing a 15-day course. Yes, I did mention Yale recovery study, which if I find my Twitter thread, I'll put it in, or actually the Meta Archive page, that's even better, on the chat. But essentially, yes, we are seeing that majority of the participants did report symptom improvement. We don't know how long that lasts. And we also saw a few patients that also developed worse symptoms. So it's not a universal sort of treatment for long COVID, but and these were vaccine naive individuals. So we had to capture them during the early phase of pandemic, but because the IRB protocol approval took so long, we only started like late in the phase of the first rollout of the vaccines. So we only had like 16 patients that we could include in that study. But the insight is quite interesting if you're interested in reading that.
Thanks. Jim Heath, did you have a question? If you can unmute yourself. And okay, we can't hear you, or I can't hear you.
Yeah, I can't.
Okay.
I wish I could hear you.
Yeah. Heather Jaspin, could you perhaps ask your question? Just we have limited time here. Thanks, Heather.
Sure. I was just wondering what your plans for the microbiome are, and I think it would be a very exciting exploration given that it seems like sex-specific microbiome may be very important in autoimmunity development, and I think could be a really nice explanation for some of your findings.
Yeah, absolutely. So we would love to do microbiome studies. We haven't collected, fecal samples from our participants. We do have some saliva samples that we might be able to, we're thinking about sequencing microbiome in the saliva, but, and we're also planning some animal bacterial transfer studies to see if there's any connection between their microbiota, the long COVID participants' microbiota in the saliva versus outcome. We are setting up a battery of behavioral tests in the animals. And by transferring antibodies from patients, this is not microbiome, but antibodies from patients with long COVID into a naive animal, we can recapitulate a few key features, few key pathologies that we see in the patient. So we're hopeful to be able to integrate the microbiome in the future, but that's not something that we are quite ahead of right now. Thank you. Jim?
Yeah, sorry, I was trying to unmute before and it wouldn't let me. Anyway,
hear you.
thank you. It was a lovely talk, Aiko.
Thank you.
I had a question. So we've been doing a lot of work on autoantibodies, and of course we're looking right, early in the disease course and on, and they do disappear after 2 or 3 months. They're just gone, more or less. But the ones we see are really a consequence of very rapid B cell maturation. They're really I think it's more of a stress response of just produce antibodies, and they're lousy antibodies. They just have cross-reactivity. And so do you have any insight into the autoantibodies that you see? Are they something similar to that? Are they just poorly matured antibodies, or are they actual, like in lupus, actual highly effective but bad news autoantibodies?
Yeah, so so we did do a panel of autoantigens that are associated with various different autoimmune diseases, and like you, there may be a transient increases in these things, but they don't last. But what we are seeing is what I just mentioned, which is that if you isolate IgG from patients with long COVID, inject it into an animal, we are seeing a significant changes in, for example, thermal sensation, pain sensation, muscle weakness is being seen, lack of balance with rotor rods, things like that. So functionally, it's there. We don't know what the B cell receptor sequences that belong to these kinds of antibodies are, but we are seeing it in a subset of patients, not everybody, subset of patients with certain symptoms that are having these antibodies. So I think there is a subset that is really relying on these autoantibodies.
Thank you.
Thank you.
Thanks. And perhaps Peter Hahn, could you ask your question?
Yeah, thank you, Jan. And thank you, Dr. Iwasaki, for the talk. I was wondering, based on kind of everything you've seen, could you comment on the cumulative risk of repeat infections? So do we see a marked difference in the risk of getting long COVID in your first infection versus in subsequent infections?
Yeah, so that type of data is most sort of prominently reported by Dr. Ziad Al-Ali's VA studies, where he found that reinfection is associated with a risk of developing long COVID. So, I would have thought that prior infection would have prevented the onset of long COVID, but that may not be the case in a subset of population. So, this needs to be kind of, so the VA data, there's a beautiful studies, but it's skewed to older male population. And if that can be sort of seen across different demographic groups, I think it will be a powerful sort of insights. So yes, repeated infection can lead to long COVID, and therefore I'm still sort of masking everyone.
Okay, thank you.
Thank you, Peter.
Thanks. And there's another question from Marco. Marco Caron, can you?
Hi, thanks so much for the presentation, the work, and also your advocacy to bring light to these issues. So my question is, so there's some biomarkers you showed and from other papers as well, where you see that there's a difference not only between those that are healthy controls and long COVID, but somewhere in between those are people who have recovered from COVID. And so there's almost a dose response relationship for some biomarkers, not all. And so could it be that what we're calling long COVID is really a discrete entity that's symptomatic, that really is capturing, in some sense, the symptomatic manifestation of dysregulation that's happening perhaps on a broader scale, so that perhaps many more people after COVID have some physiological dysregulation, but only some get called long COVID. And I guess this is somewhat related to Peter's question, in that perhaps then repeated infections would push more and more people towards this sort of symptomatic category. So what's your take on this? Is this the tip of the iceberg that we're seeing when we call individuals long COVID patients?
Thank you, Marco. Excellent question. So essentially, long COVID, there's not a unified clinical diagnostic criteria for long COVID. CDC definition, WHO definition, if you use them, anyone who has new or ongoing or exacerbated health issues after COVID would have long COVID that lasts for over 2 months, right? So by that criteria, I think a lot of people would have long COVID. Fortunately, if you've only had it for a couple of months, that group of people, there are many who will recover from that. But unfortunately, if you look at 1 year, 2 year, 3 year post-infection, and if they're not recovering, that's sort of the syndromic long COVID that's just very difficult to treat. And some people get worse during these many years of chronic syndromes. So, first is that there isn't a great definition that's unified to characterize long COVID. And second is that long COVID is likely consists of multiple different endotypes. And depending on which one you have, not only the diagnosis and therapeutics will be different, but also maybe the recovery is also going to be different. And third, I'd like to point out that what you said about the awareness of long COVID and how people are sort of self-recognition of long COVID is very variable across the globe. So in places in South America, for example, long COVID is not really at the forefront of any media coverage. Many people have never heard of it. And in those situations, people are suffering with long COVID, but without recognizing it as being something that occurred after the acute phase of COVID. So there's a lot of inequality among different population in awareness, diagnosis, treatment, all these things. It's just a big mess out there.
Thank you.
Thank you.
Well, I want to ask one question, and then there's still some other questions, but they're repeat questions. So as a pediatrician, I am very interested in the fact that we just don't see it, especially in kids. I don't see it, or we don't hear about it much, especially in kids who haven't reached puberty. So I was interested in your hormonal regulation issues. Have you done age-based studies, and do you have data? We see it perhaps I see it in the older teenagers much more than I do in the younger teenagers. So could you talk about age, and particularly young age?
Yeah, absolutely. And so that's been the sort of what I'm seeing in reports, as well as our own. We are doing a scoping review of pediatric long COVID right now. And what you said is exactly right. We're seeing a lot more. First of all, it's not as much as what we're seeing in the 30 to 50-year-old category that I mentioned before. But we are seeing a significant long COVID cases in patients that are in their late teenage group. And there seems to be female dominance there as well. So that's sort of holding up across different reports. And we're also seeing that in very, very young children. Again, it's very hard to monitor these things and measure them because they may not be able to articulate their symptoms, but that's not the group that we're seeing a lot of cases there. So yes, the hormonal, that's why I think the sex hormone is going to play a major role in the onset as well as regulation of long COVID.
Thank you. And let's see, there's another question from Wes Van Voorhees? Wes, can you ask it, or I can
What
ask
was
it?
answered? Thanks.
Oh, it's answered. Okay. And then I think we have time for one other question, and Michael Davis has another question. Can you ask that?
Yeah, I can. It was just a quick question about the mouse studies that you were talking about. I was just curious whether or not the mice needed to stay within the ABSL3 space the entire time, and what the duration on those studies would be.
Yeah, so with the antibody transfer study, that's a BSL-1 study, thank goodness. But for the COVID, SARS-CoV-2 infection plus something else, that's where we have to do a BSL-3 study. And we are remodeling our facility to be able to do some of the measurements of the behavior, but it's very bulky, and we can't do all the measurements of behavior in that facility.
Wow. Well, thank you just so very much for your really interesting, stimulating, and eye-opening talk. It was so clear, and we just appreciate all of us online. There's a lot of us online who have enjoyed it. I would like to thank as part of the symposium organizers, we are so grateful for presenting to us, and thank you very much. I want everyone to know about the February 1st next symposium, and now I'll be quiet. Yes.
Thank you